Donc, j'ai grand plaisir à présenter Morgan Craig, qui est, comme il était mentionné, professeur adjointe au département de mathématiques et statistiques. Donc, Morgan a fait son baccalauréat au Collège Saint-Boniface. She did a master's at the University of Manitoba and then a PhD in pharmaceutical sciences at the University of Montreal. And she then was a postdoc in evolutionary biology at Harvard University. And then, elle a été embauchée comme prof adjointe au département de mathématiques et statistiques de l'Université de Montréal. Elle a obtenu de nombreuses bourses du CRSNG, ce qui fera plaisir à un de nos invités. Et elle a aussi obtenu le prix Lee Siegel de la Society for Mathematical Biology for the best paper in the Bulletin of Mathematical Biology. So I'm very pleased to introduce her, and I would give her now the...
So thank you everyone for coming on a Friday afternoon on a hot day in the summer for this talk. I'm very appreciative of the invitation from the CRM and from Luc Finet and Jean-Philippe Lessard and from all of the people who spoke before me, and to be a part of this initiative of the expert team in epidemiology and public health activities. So I'm going to talk today about the work that my lab has been undertaking to try and understand the immune response to SARS-CoV-2 infection. And just to give you a bit of background, my lab focuses on applying and implementing quantitative approaches, particularly computational biology, to study new ways to treat a variety of diseases, particularly immune disorders and cancer. And so we've taken some of our methodologies and applied them to try and understand the in-host dynamics after infection by SARS-CoV-2. Oh, there we go. So as was mentioned by the people at the beginning of this talk, epidemiological models of transmission are critical components of public health decision-making and planning. And we're all used to now hearing about the models and what are the models predicting, which is kind of scary as mathematical biologists, because if we get it wrong, we may never get funding again. But within these epidemiological models are a class of models that most people are probably familiar with, called SEIR models. So susceptible, exposed, infected, and recovered individuals, and looking at the transmission between them. So my lab is focused particularly on the infectious portion of this cascade. We're trying to understand how the virus, after it enters the body, elicits an immune response, and how that immune response can be determinant for the severity of COVID-19 presentation. And so I'm going to talk, there are going to be a lot of different projects that come in here. I'm going to mention cancer, but all of these different facets of the research that we do in my lab have contributed to our ability to mount and construct these mathematical models, these quantitative approaches with which we can interrogate SARS-CoV-2 infection in the host. So to give a bit of background, here this is the SARS-CoV, so the original virus of SARS, its life cycle, and not the current one that is circulating, but it still gives a good idea of how the viral life cycle works in host cells. So the virus enters your body primarily through respiration and also through mucus transfer, and then enters into the respiratory tract, where it is a single-stranded RNA virus. So the RNA is contained within the envelope protein, and there are spike proteins around there. Those spike proteins bind primarily to what we think are ACE2 receptors, though others may be involved on the surface of epithelial cells and other host cells. And once the spike protein binds to these ACE2 receptors, the virus enters the cell through what's called endocytosis to begin replication and its copying process. So after entry through endocytosis, viral RNA contained within the envelope is released, and this released viral RNA is transcribed into structural and non-structural proteins, which are repackaged into virions, which is one viral protein, and then they are released from infected cells through exocytosis and also potentially cellular lysis if they are destroying the cell. And so this process is integral to the steps with which we approach modeling the immune reaction. And to do this, we use standard techniques that have been used in studying viral infections, including HIV, which I'll talk about in a little bit. And so this is a viral dynamic model of how this viral replicative life cycle is occurring within host cells. So you have uninfected cells, so susceptible cells like in the SEIR model that I talked about just before, that become infected through viral proteins. And then these newly infected cells undergo a time that's called the eclipse phase, before which productive infection occurs in the cells. And before becoming infected cells. And then these infected cells die eventually and also are shedding and releasing more viral proteins into the environment. And those viral proteins are going to infect new cells, and the cycle continues until the immune system comes and helps you out, hopefully. And so in chronic infection like HIV, we typically include production and death terms of these susceptible cells. In acute infections like SARS-CoV-2, we may not include those. However, I'm just going to mention that in our model, we do, because as I'll talk about in a little bit, we're hoping to study differential response in COVID-19. And so we want to see whether we can get back to homeostasis, so back to stability in the system, in the host. And so here I'm just showing the eclipse, the viral dynamics model that we are working with, or the reduced model. So we have susceptible epithelial cells, which we allow to regrow because, as I said, we're looking to get back to homeostasis in the system. Hopefully the person recovers. After infection, these cells go through the eclipse phase before they become productively infected cells. These infected cells die at some death rate, and then they produce before death viral proteins that go and infect new cells. And so I'm showing here data from macaques. Other teams have also worked on data from humans, and just showing that this model recapitulates the viral load over time in infected non-human primate models. Now, the thing that's important to note here is that this model does not include the immune system. And so there's this viral clearance term that takes care of that, in the sense that here, obviously, these primates did have functional immune systems. And so we're seeing viral load here, and that is a virus that is also encountering an immune response. And right now, there's nothing to model this immune component. But as we know, of course, there is an immune system, and hopefully it works in a way that is not dysregulated to produce an effective control of the SARS-CoV-2 infection once infected. We know that there's significant involvement of the immune system after infection, and that severe COVID-19 presents with hyper-inflammatory symptoms that are not necessarily linked to actual viral load or the current viral load. So here in the top left, you can see again this viral life cycle of the SARS-CoV-2 infection, endocytosis, replication, and exocytosis. And once epithelial cells or other cells, depending on your virus, are infected by the virus, they begin to secrete a host of cytokines, which I'll talk about in a little bit, but are signaling molecules to signal to the immune system that, hey, something's wrong, you should probably come help me out. And so these host of cytokines are secreted. And as you can see here in the schematic, and I'll talk a little bit more about this in a little bit, immune cell subsets are attracted to the site of infection. And if these immune cell subsets work in the way we would hope, these alveolar macrophages, so resident macrophages inside of lung tissue, can learn to recognize the virus and also recognize infected cells, and you'll get priming of CD8 T cells and CD4 T cells that can also work to apoptose the infected cells and infected cells may present antibodies that also help the immune system effectively control the infection. However, as you've probably heard in a severe COVID-19 presentation, typically patients exhibit a lot of cytokine storms, so this hyper-inflammatory reaction, uncontrolled and dysregulated immune response. And this dysfunctional immune response can also include a number of other immune cell subsets that inhibit essentially the role of T cells to be able to tamp down the infection. And so this is recapitulated in data from patients. An emerging characteristic that defines mild and severe COVID-19 is a differential immune response with respect to the cytokine storm that I just mentioned, and also hyperinflammation. And this includes elevated levels of interleukins, which are specific cytokines that promote the development and differentiation of T and B lymphocytes and other hematopoietic cells. And particularly, this translates into a lymphocytopenia, so a lack of lymphocytes that you see recapitulated here, particularly in T cell subsets. And so though some of these damaging cells like neutrophils, if you have hyperinflammation, you may have a lot of neutrophils that stay around for a long time. It doesn't appear like neutrophils are particularly increased in severe COVID-19 presentation, but due to the lack of lymphocytes in the blood, there is a high neutrophil to lymphocyte ratio, and this is indicative of severe COVID-19 presentation. So lymphocytopenia, high NLR, and also this hyperinflammation and cytokine storm. And so my lab has worked in particular on understanding immune cell and blood cell production and function. And so here I'll take a step back and just describe a little bit about how the immune system is orchestrated to give you a step forward into the modeling that we're doing right now. So blood cells are all produced from hematopoietic stem cells in the bone marrow. And hematopoietic stem cells, like all stem cells, are capable of self-renewal, so they maintain their population size. And eventually, they differentiate into a number of different cell subsets, including, of course, your red blood cells that produce oxygen, platelets, which will clot and are implicated in some immunity, and a host of white blood cell subsets, either involved in the innate immune response, so really kind of the foot soldiers of your immune system that arrive on the scene quite quickly, or the adaptive immune system that are primed through antigen presentation and such. And all of this hyperproduction of blood cells occurs through signaling through cytokines, which I've already mentioned. And so your body is producing about 100 billion blood cells a day, which is an insane amount of cells to me. I don't know if that blows your mind, but it blows my mind. And all of this is assured through robust feedback mechanisms with signaling proteins that are called cytokines. And these cytokines are very small proteins, or small proteins that act via the association to cell surface receptors to drive differentiation and production and maturation of blood cells, and also prime them for immune function and inflammatory response. They can also act as chemokines to recruit cells to the site of infection, and this will be particularly important in the modeling that we're going to do. So some of my early work was really focused on understanding the GCSF and neutrophil axis, so the production of neutrophils mediated by GCSF. As you can see here, GCSF has some effect on other immune cell subsets like monocytes, which I'll talk about in a second, which are another type of myeloid cell in the innate immune system. And they can also have an effect on dendritic cells that act to prime T cells in the lymph nodes. But GCSF's primary action is the production, maturation, egress of neutrophils out of the bone marrow. And it does this in a variety of ways, which I will summarize right now. So this is a kind of schematic model of how GCSF and neutrophils are produced via GCSF. So as I mentioned, neutrophils are blood cells, so they're produced by hematopoietic stem cells. They undergo a period of proliferation in the bone marrow. And what's happening during this period is that they're exponentially, they're growing in number. So there are a multitude of doublings that happen during proliferation. And then neutrophils stop dividing altogether. and mature in the bone marrow, where they grow in size, and they also grow in number of receptors on their cell surface, and particularly GCSF receptors. And mature neutrophils are then sequestered into the bone marrow reservoir, from which they egress into the circulation. And from circulation, neutrophils frequently become marginated within the tissues and the capillaries in the tissue. And the reason that there's this large bone marrow reservoir in the bone marrow, obviously, is because there are, neutrophils are very short-lived white blood cells. They have a half-life of disappearance from circulation of about 7.5 hours, maximum 16 hours or so. And so there are about 10 times the amount of neutrophils in your bone marrow able to be quickly recruited into circulation. And this is important when you're thinking about acute infection dynamics. Oops, sorry, I went the wrong way. So these actions that I just described are all mediated by GCSF. So the primary action of GCSF, whether you have falling neutrophil numbers or whether you have some infection that is an infected cell secreting a chemokine and recruiting cells to the site of infection, is to modulate the release of neutrophils out of this bone marrow reservoir into circulation. And then GCSF acts upstream to restock this bone marrow reservoir to make sure that in the case of another acute emergency, falling neutrophil numbers, infection, you'll have enough white blood cells, particularly neutrophils, to respond. And the reason that we originally embarked on this was because we were studying myelosuppressive chemotherapy, which is chemotherapy that's cytotoxic to cancer cells, but also to neutrophils. And so this is a particular way that even though we were working in cancer, this model can translate into the acute infection dynamics like I'm going to describe. And because chemotherapy kills dividing cells and proliferative cells like white blood cell progenitors, we also have extended this model to include monocyte production from the bone marrow. This was work by my former PhD student, Tyler Cassidy, who's now at Los Alamos working with Alan Perlson, probably on something similar to this. So after proliferation, I already explained that some of these neutrophils these neutrophils start to mature, but some of these progenitors also become monocytes in the bone marrow. These monocytes don't have a reservoir, so they divide once in the bone marrow and then egress into circulation. And monocytes can become macrophages, and that will be important in the infection dynamics that we're going to look at. So just to show you that this model can fit very well to the dynamics that we see in chemotherapy, and it will fit very well, or hopefully, as I explain, in the infection dynamics that we're going to see in a second. So all of these actions are mediated by cytokines, and sorry, I'm just going to take a sip of my water. Okay, I meant to put that on mute so you didn't have to hear my water bottle. So all of these actions are mediated through the association of cytokines to receptors on the cell surface. And oops, the way this is normally modeled is through Hill function dynamics or Michaelis-Menten binding kinetics. And so here I'm just showing you a little bit of what this is based upon. It's a chemical reaction between substrate and enzymes. And these substrates and enzymes are associating to each other and forming a bound product. And this, through the assumption of equilibrium, can be modeled using this Michaelis-Menten binding kinetics, where you have some maximal rate that this reaction is occurring at, and a constant that accounts for the half maximal, the concentration at which half maximal speed of reaction occurs. And what is crucial here is that this is occurring through the assumption of equilibrium. And so when we started modeling GCSF dynamics and neutrophil dynamics, we leapt off with this model where we assume that there's equilibrium between bound and unbound GCSF. and that this was occurring through this Hill function, which is similar to the Michaelis-Menten function I just showed, but with a power here that denotes the slope of the line. So we leapt in with an early GCSF model that is typical of pharmacokinetic modeling. And here you'll see that there are two primary routes with which GCSF is cleared from the body. The first is renal elimination, which is a linear elimination term. It's essentially your kidneys working to remove small proteins, these cytokine proteins. Also through neutrophil-mediated elimination, which is how your body is generally getting rid of GCSF, which is that it's associating to receptors on the surface of neutrophils. And the reason this is important, and you see a dose here, is because during chemotherapy, GCSF is given exogenously as a drug to rescue falling neutrophil numbers as they're being killed by the chemotherapeutic agent. But unfortunately, this model wasn't shown to fit very well to the data when we started modeling it. And so we realized that what was happening is actually this equilibrium hypothesis wasn't holding true, and that the proportion of neutrophil-mediated elimination was much too low to capture this dogleg in the data. And so we constructed a new model that accounts for the unbound and bound concentrations of GCSF. And you'll see here now that there are two routes of elimination as before, but both of these eliminations are now linear, and that there are nonlinear terms in the binding and unbinding of GCSF acting upon the neutrophils. And if you make the same equilibrium hypothesis as in the Michaelis-Menten model that I showed earlier, you recapture the Michaelis-Menten or Hill function dynamics that we would expect. So essentially this is saying that at endogenous concentrations, it's very important to to obtain accurate parameter estimations of how much neutrophils or GCSF is binding to neutrophils. It's very important to take into account the binding and unbinding dynamics. And so with this updated model, you can see here that it does a much better job of capturing this kind of dogleg in the data. And I mentioned all of this, it seems like a sidetrack, but actually it becomes very important, and it's kind of the launching off point for the model that we are one of the models that we're working on for studying SARS-CoV-2 infection. So we model all of the interactions between the blood cells that will be included in our infection dynamics model and the cytokines that are mediating their recruitment, their function, and their production, proliferation through this bound-unbound cytokine model. And so in a generalized form, again, I'll just kind of go through what's going on here. You have cytokine that is produced endogenously, that free cytokine that is cleared through kidney clearance or renal clearance, and then a binding term that produces bound cytokine, and an unbinding term that reproduces freely circulating cytokines, the cytokine that you will measure in a patient's serum. And then once bound, this bound GCSF or cytokine can be internalized and can also unbind, as I mentioned. And here you'll just note that because this is now generalized, we take into account any kind of target cells. So I mentioned earlier that there are robust feedback mechanisms of cytokines in the blood cell production system. And the reason for this is so that if you are missing one cytokine, you hopefully can still produce some of your blood cells. And so, for example, GCSF can associate to myeloid cells, so monocytes, and also primarily, of course, neutrophils. And so this model can take into account the different immune cell subsets that are affected by the cytokines. And so this is now important because if we return back to trying to understand the immune response in COVID-19 and after infection, we know that cytokines play clearly a role in determining the severity of disease presentation. And in particular, it's been shown for SARS-CoV, the original strain, and also MERS, Middle East Respiratory Syndrome, that interferon 1, so type 1 interferon signaling, is very important and almost determinant in some sense for disease severity. So if you have early interferon 1 signaling, or if there's a lack of interferon 1 signaling, but particularly this early and delayed, let's look at this, if you have early interferon 1, you have a good control of the virus, you have a regulation on the amount of inflammation that is presented in the individual, and you have mild disease. But if you have a delayed interferon 1 response, you see here that there is an increase in viral titers, there is increase in lethal pneumonia, there's an increase in immunopathology, and there's also an increase in the hyperinflammation that you see in patients. And so we are studying, or this is part of the component of what we want to study, is what are mechanisms that determine this early and late interferon 1 signaling. So here we are. We finally arrived at this slide that talks about the immune response to infection. So this is a model that we're working on, particularly my postdoc Adrienne Jenner and Sophia Alfonso, who's a very talented upper level undergraduate who I work with at McGill. And this is in collaboration with Rosie and Amber at the University of Tennessee, Rosie Ayogo and Amber Smith at the University of Tennessee, and Courtney Davis at Pepperdine University. And so you'll see here that we have a model for the viral dynamics that I showed earlier. You have virus and susceptible cells that become infected through viral interactions that then become dead cells. And now we have a host of immune cell subsets that have showed up to model how the viral loads are changing, and also what the immune response is to infection. So along the arrows are kind of the mediators of the response. So obviously susceptible cells become infected cells through viral interactions. Here we also include the macrophage subsets, so resident macrophages in the lung tissues and the inflammatory macrophage subset. Macrophages are particularly implicated in phagocytosing infected cells not infected cells, dead cells, and clearing up debris after infection. Once cells are infected, they secrete interferon that then recruits these immune cells onto the scene and mediates the immune response to infection. And again, I'll just reiterate that certain ones of these cell subsets are producing a variety of these cytokines, for now a reduced set, but as we get more data from patients, which I'll talk about in a second, we will also include different cytokines and immune cell subsets. And these cytokine models are, as I just mentioned, the unbound-bound interactions with immune cell subsets. So this model can recapitulate healthy control of infection. So here I'm showing again, this is kind of small, I'll just take you through it. These are the viral load dynamics now in an individual and a human, and using the parameters that we had fit from macaques. And you can see here that in red are the susceptible cells that obviously are quite decreased as viral loads increase, as infected cells increase, but are able to rebound once the immune system has tamped down and controlled the infection. And so you see that this recapitulates grossly what we would imagine is happening, or not imagine, that we know is happening in infection. Inflammatory macrophages rise through conversion from monocytes and also from tissue-resident macrophages. Neutrophils are recruited into the scene of infection and stay actually quite high, which is what Amber Smith, who also works in a wet lab, observes in animal models of infection, that neutrophils stay elevated past 10 days, etc. We also see the delayed arrival of T cells, which is normal because T cells are primed in the lymph nodes by dendritic cells and then arrive on the scene of infection. And here you can see the dynamics of bound and unbound cytokine concentrations that are acting to regulate all of these immune cell dynamics. So this is basically, of course, hopefully I should have said this before, this is all work in progress. We're still working on this, but we have a working model now of the immune system response to infection. One component that I haven't yet talked about is that within the host, this immune cell pressure exerts, or this immune cell response exerts a selective pressure on viruses, which causes them to mutate. We also know that viruses mutate after treatment, like for example in HIV, and to become resistant. This selective pressure causes changes in the fitness landscape, which you'll see kind of in these bumps here of the virus. And the virus then seeks to mutate and adapt to escape the effects of the immune system, or potentially a treatment that, like an antiviral. And so to account for additional strains, the basic model of viral dynamics can be adapted to track mutant strains as they arise. And this is based on the probability of mutation at specific spots along the viral genome. So if we're thinking about HIV, for example, we know which proteins under drug therapy will mutate, and we know which nucleotides are coding for these proteins, and we can back calculate what the probability of mutation is at that specific spot in the genome, given the change in protein that we observe. And so here is just that viral dynamics model that I showed earlier with wild type virus, but also now taking into account potentially the emergence of new viral strains. And so this is something that we had worked on previously. This is part of what I was working on in my postdoc, where we used a quasi-species model for HIV and combined with different drug combinations that are used in patients, or to treat HIV in patients, and the resistance pathways to understand and predict therapy outcomes, so versus thinking about viral load and genotype over time. And in particularly, we applied this model to understand novel ways of delivering antiretrovirals. So instead of having somebody take a drug every single day, our collaborators at MIT had developed a gastric retentive device that looks like a pill. This is not to scale, it's not this large, but this pill you swallow like you would swallow any normal pill, but it opens up into the star in your stomach. And there's drug loaded in each of these arms. These arms are made up of polymers that degrade in the stomach and are linked by an elastomer core that also degrades. And as you can see on the right, instead of the rapid rise and fall that we see with commercially available antiretrovirals, so the antiretroviral or HAART that you would be taking, or patients would be taking now, or individuals with HIV, sorry. The gastric retentive device keeps sustained levels of drug over a week. This is thought of as a weekly dosing of antiretrovirals or HAART. And so we had applied our model, just to show you that this works, this quasi-species model. We'd applied this to understand how the introduction of a weekly oral gastric retentive, like long-acting device, compares to what is available on the market right now, and showed that it would be equally as, you would provide sufficient coverage, equally good coverage as what is being used right now. And so the reason I bring this up is because together with Julie Hussein's lab at the Montreal Heart Institute at the University of Montreal, who were working to incorporate this idea of viral strains on the in-host model to understand the interactions between the virus and different viral strains and the immune system. So it's clear, obviously, that understanding how viral strains appear and are spreading is critical to predict the epidemiological evolution. It's also critical to understand how those strains are appearing in the body, so that we can potentially use targeted therapies to stop that, or to at least know and predict how they might appear. Julie works on inter and intra-population genetics of SARS-CoV-2. This GIF that you're seeing right now is her lab's analysis of over 28,000 sequences. This is transmission, so this is more epidemiological, or the appearance of strains. It's different strains that are defined by 17 different viral mutations, which are labeled on the edges of this network, and that are found at 5% frequency worldwide. And the map shows the relatedness of different strains and their frequency in different continents. And the animation is reflecting cumulative frequencies with a jump of 10 days, last updated on May 18th, 2020. So you can see that different strains are spreading from in different regions. And we'd like to understand why this is, and if, firstly, how that affects on the immune response, but also how the immune response is making these viral strains arise. And Julie has already started, and her team has already started to look at intra-host dynamics, and we're hoping to have more data for this as we go on, or we know we will, and it will just take some time. So with our student Viv Crow at Concordia University, we are working also to translate from Julie's strain-based population genetic analysis into the viral dynamics model, to understand, as I said, how the immune system selects for given strains and how strains differentially affect the ensuing immune response. So this is kind of a large project. As I said, it's still work in progress, but we can recapitulate infection dynamics. And in parallel, we are also working on a localized tissue level model. And so I'm just going to talk briefly, take a step back again into cancer, but just to explain where this project came from. So in parallel to the systemic model, like I mentioned, we're working on tissue level dynamics. And this is done through agent-based modeling. So in case you may not be familiar, I'm not sure, agent-based modeling essentially assigns different cell types to be agents. I mean, it doesn't need to be cells, they can be individuals. They're agent-based models for epidemiological spread. And in our case, we had been working on a model of glioblastoma, which is a deadly central nervous system tumor, and has a very, very low survival rate of 5% over 5 years. And so to understand how we can better treat glioblastoma, we're working with cancer biologists at McGill and at Harvard, looking at immunotherapies. And one of these immunotherapies is an oncolytic virus. So it has already a viral dynamics component in it, because oncolytic viruses are genetically modified viruses that infect tumor cells preferentially, causing them to die, but also to elicit an immune response. And so in an ABM, you have different cells that interact via different rules. And these interactions dictate from time step to time step how where they might be positioned in space, and also what their behavior is as time progresses. And so you can see that what a screenshot of an ABM of glioblastoma or whatever might look like, where you have, instead of a homogeneous mix of, like we have in the systemic model, you have really individual agents that are operating over space. But you can extract the same kind of information that you might get from a systemic model to kind of map from the localized level into a systemic kind of homogeneous population model. And so the kinds of things that we had been working on, and this is again with my postdoc Adrian, is to implement in a glioblastoma model, particularly the immune cell response, and also this oncolytic viral therapy. And this is done using a computational framework called PhysiCell that was developed by Paul Macklin at Indiana University. And Paul jumped in, realized how effective PhysiCell could be to understand tissue level dynamics of SARS-CoV-2 infection. And so he launched and leads the SARS-CoV-2 Tissue Simulation Coalition, of which we work in collaboration with him, in also collaboration with Amber and Rosie and Courtney, who I mentioned earlier, who work on the systemic model. And so here you can see, I just showed you, if you're interested, you can go to the website. It's a large international coalition that's focused on developing a comprehensive multi-scale framework for SARS-CoV-2 infections in the lungs and gut. And this framework can then be adapted to suit the individual needs of each individual research group of the collaborators. But the idea is that we're not, reinventing each research group isn't doing the same thing. Let's pool our resources together to rapidly get this model to develop this model. And so we work on a rapid prototyping system. I've put the link here. I think this will be posted later, but you can also get it from the website, where we release a preprint every 2 to 3 weeks, and we're constantly doing new versions of development in a variety of phases to scale up this tissue level model. So the last release that we have, V3, version 3, should be out shortly, but I'm showing you here version 2, which does not include, as of right now, an immune component, but does include the viral dynamics that I explained earlier, but on a localized level. So you have viral particles that are infecting individual cells, and then all of the components of viral replication, well, endocytosis, viral replication, and exocytosis that are occurring within infected epithelial cells. And what's great about working with ABMs is you get really fun little simulations that you can show instead of just static images. So here is, as of V2, I mean, obviously we're not at the point where this is recapitulating infection dynamics because everything dies, but it is, as I said, a scalable framework that will be applied in a variety of different research initiatives. So here, just to show you, because we'll need this for later, blue are epithelial cells that become infected when they turn yellow, or they become infected and turn yellow. Gray, they then undergo viral replication within the cells and turn gray, and black cells are dead cells. And so the next phase of this, so the way that we work is that we have a large coalition and we have individual submodels, including an immune response, a damage response, a lymph node team. We have a viral replication team, for example, and Adrienne, Courtney, Amber, and I, among a lot of different colleagues, other colleagues, are working on the immune response, which will be part of the version 3, which should be out shortly. And I'm showing here something just to give you an idea of the flavor of how the immune cell, how we have to think about the immune cell now, not at the localized or systemic model of, you know, we have recruitment from the bone marrow, etc., and we have systemic cytokine concentrations that we can measure like we will measure in patients, but actually now we're looking at how do these immune cells move within space? How are they interacting with infected cells? So on the left, you have only macrophages interacting with infected and susceptible cells and dead cells that are black. In the middle, you have only T cells. On the right, only neutrophils. And they work, they are recruited and along chemokine gradients into wards infected, the site of infection, and then they work to phagocytose and to apoptose of their T cells, infected cells. And so this is, like I said, a very early prototype. It's not the last version of the immune response, but this is now, we're at the point of finishing the version 3 cycle, and we'll be working version 4 to improve and include more cell subtypes. So I only have a couple more slides left. Just to recap, just to conclude a little bit about what our lab is doing. So we have calibrated a viral dynamics model of SARS-CoV-2 infection at the systemic level, which is integrated into a mechanistic mathematical model of the systemic immune response that we are working together with collaborators to include different strains and their impact on the in-host model. And we also in parallel work on an immune submodel of the larger computational model of infection dynamics in tissues. Now, this has to go somewhere. We will be getting a large amount of patient data from the hospitals in Montreal that will be quantified via FACS and ELISA normal standard techniques. And our clinical collaborators and us, we are also are interested in identifying the characteristics, like I mentioned at the beginning, that distinguish mild versus severe COVID-19. We're particularly interrogating on interferon dynamics. And we can also, with these models, our models, test potential therapeutics. And just to give you an idea of how we do that, with my former PhD student Tyler and Adrienne, we've developed an in silico clinical trial platform in my lab. which basically takes a disease, some pathophysiological mechanisms, and builds a mechanistic model like the ones I've shown you here today. And these models are always built from existing literature data or from data from our clinical collaborators. And from this model, we can expand and generate cohorts of virtual patients that we make sure respond in a physiologically relevant way. So here there's a Kaplan-Meier curve. We just check to make sure that we can recapitulate clinical trial dynamics, etc. And then we can run an in silico clinical trial by looking at different drug combinations, looking at new novel potential therapies, trying to understand ideal and optimized therapeutic schedules so that they can be implemented either at preclinical development stage or by our clinical collaborators who will be feeding us data and want some predictions back. And so just to wrap up, using this in silico clinical trial platform, we're going to work to understand mechanisms between the differential immune responses in COVID-19, particularly because I'm a researcher at Saint-Justine. We want to understand how age affects what is the immune system dynamics, how are they differentiated between younger patients and older patients. Usually we're looking to understand how in-host dynamics cause different viral strains to emerge, and also between our two tissue level and systemic models, understanding how local tissue level dynamics contribute to systemic observations. And then ultimately the goal will be to also test proposed therapeutic interventions, including stratifying patients as responders or non-responders and establishing effective therapeutic schedules that can be used to treat individuals. Like, for example, how early do you need to give an antiviral drug? Do you need to modulate ACE2 inhibitors? Those kinds of questions. So I hope that you got the impression that this is not something that I do all by myself, of course. So from my team, Adrienne, Sophia, and Viv have been working very hard this summer, especially Adrienne, to construct these models and get them up and running. And we work with a large number of collaborators, Amber, Rosie at the University of Tennessee, and Courtney, and Judy and Guy at DMS, and also other collaborators that are looking at sequence evolution in host, and also the SARS-CoV-2 tissue simulation coalition that, of course, I have not listed everyone because it's a very large group of people. So thank you very much. Again, thank you to the CRM and to everyone for coming today, and I hope that has opened the floor for questions. Thanks.
Indeed. Thank you very much for quite a tour. So I didn't realize your weeks had more than 168 hours.
Yeah.
This happens to some of us, I guess. So any questions that you may convey through the chat if you want, or if not, I will ask you what everyone is asking, anyone who's doing, oh, here's one. So I have a question from Jessica Conway.
Hi, Morgan. Great talk. Thank you for that. My sound actually cut out during part of your talk, so I have a really nice softball question for you.
Great, thank you.
You said, you're welcome. You said the models were validated with data. I just missed what the data was.
Right, so for right now, we're using a variety, so it's actually a large variety of data. The way that we went about it is by doing kind of like submodels. So we have literature data for how these cytokines, for example, modulate function, modulate proliferation. We also have data from animals, and we will be getting data from actual patients. So so particularly plasma concentrations of immune cell subsets and quantification of cytokine concentrations over time from patients. So we're kind of in the middle phase right now, where we're integrating via existing literature, and then we'll be integrating a large amount of data from hospitals around Montreal.
I guess I'm confused. So thank you. But literally the curve I saw, what were those dots?
Oh, which, the viral dynamics one?
Yeah.
Oh,
Well,
sorry, I wasn't sure which one you were talking about. That is data from macaques. It is a paper in Science.
Okay, so you're using a mixture of, and the immune aspects are validated using human data in the literature?
That's right. And we also have access to viral dynamics data from humans. I just showed the fit to macaques, but that component can be redone to human curves. Right now it's done to non-human primates.
But so far you don't have, which this would be incredibly difficult to get. This is not a judgment. I just want to be clear. You don't have like a global, like all the measurements from the same person.
No.
Is that likely to happen?
Sorry?
No, dream data, is that likely to happen with the current clinical trials that you're really working with?
Yes, potentially not viral load, but
well, that's not really a thing for this.
Exactly, right. So, but yes, for immune and cytokines.
Cool. Okay, thanks.
So I have another question from MD Afsar Ali.
Hello, can I ask a question that is, thank you for your good presentation and excellent research. I have a question that is here, cytokine is proportional to infected cell or virus? How did you calculate that is cytokine?
So cytokines are actually a function. It's a functional response. So it's not just necessarily proportional. They're nonlinear and they're generally Hill functions. And these were all determined based on existing data for how from other similar. So if we don't have access to SARS-CoV-2 infection data, we use SARS or MERS. But we also have a variety of data sources from other viral infections are also in other scenarios that we use to calibrate those curves.
Okay, thank you.
Thanks.
There's another question from a Professor Anthony Humphreys. So Tony, you can ask your question.
Yeah, if I can unmute myself. Yeah, so several of the questions are getting into the details of this model with the cytokines, which, as I understand it, you haven't published yet, Morgan. When can we expect to see that in a preprint?
Of the systemic model? In the next couple weeks, max.
Oh, cool. Look forward to reading that.
Thanks. But the generalized, so just because this is Tony who wrote this model, the cytokine model is based off the model in our BMB 2016 paper that we published with Michael Mackey.
Okay. And the next question from Kathleen Wilkie.
Yeah, hi. Thank you very much, Morgan. I really enjoyed your talk. I was wondering if you could speak to the patient-specific response and how you have thought about trying to get to that. Because that seems to be like the big crux is how do we know if we're in line for this severe reaction or not? And you had that nice curve showing that it might depend on early IF and gamma response. I'm just wondering with the data that you're getting, are you able to differentiate what goes on early on to predict whether it will be a severe reaction or a mild reaction?
So this is a great question. So it's kind of twofold. There's kind of two ways to think about it. So just to be clear, we don't have this data yet from patients. We're still waiting for that. But there will be two kind of ways we can attack it. One is once we have the data, we'll do some like nonlinear mixed effects modeling to stratify patients and look for in the actual data what differentiates them, for example, and then simulate our model or use our model to see what needs to be changed, for example, from the healthy infection, that's not really what I mean, but the controlled response versus the uncontrolled response. So what needs to be modified such that we can observe the dynamics that we see between these patients. And also because we're interested, like I mentioned, I'm very interested in age-related responses. We know there's existing data, this is known, how immune cell, or somewhat known, how immune cell response and function and production changes with age. And so we will incorporate functional responses based on patient age into the models so that we can then look at what is actually dynamically changing between different patients of ages or you know, existing comorbidities, expression of ACE2 receptors, etc.
Okay, and so I will take one last question from Shigi Ruwan.
Hello?
Yes, go
Hi.
ahead.
Yeah, Morgan, yes. As we know that we do have two types of cases. Some are very severe, some are mild. So certainly I think the responses are slightly different. Do you consider different data? Do you consider the data the same and expect, estimate the parameters for general or you know what I'm talking about, right?
Yeah. I hope I do. Let me know if I didn't answer this correctly. So for right now, we've so everything, the parameters are estimated. This kind of actually relates to the question that Kathleen just answered. All of the parameters are within the kind of normal range that we might that we've observed in existing data, and then depending on the data that we get from patients, will likely work kind of back. So what needs to be changed, which parameters need to move in a certain way, or which way are they moving, so that we see the qualitative and quantitative dynamics that we observe from patients. So we're not going to, we didn't do a bulk estimation of at one of the whole model because there are too many components, but each of the kind of divided into submodels were estimated that way. I'm not sure if that answers your question.
Yeah, but because from the literature I have read, seems that from some of the severe cases, so for example, the response from the CD4 and CD8 reactor are not as active as the others. You know?
Exactly. It seems like in hyper-inflammatory scenarios, the T cell response is quite dampened, and they actually might be killed by damage, essentially. So yeah, that's something that we will be looking at.
Okay, thank you.
Thank you.
Okay, so thank you very much all for attending, and thank you very much, Morgan, for this very nice and challenging presentation in a positive sense. So, merci beaucoup, et je souhaite que les prochaines éventuelles rencontres ne se fassent pas dans ce format, puisque le groupe va à l'évidence trouver une solution au problème du COVID-19. Merci beaucoup à tous.
Merci.